Eukaryotes, some prototis, they have cell wall. And some fungus, they have a cell wall. And plants, they have a cell wall. Animals, no cell wall. That means a cell wall is an important component. Most of the organisms, they have a cell wall. So we are interested at this point only a cell wall of prokaryotes and cell wall of plant, cell wall of bacteria and cell wall of plant. Why we are not interested prototis and fungi cell wall? Because the learning of certain things has a use. If we learn all these things, at the end they are not much useful. It's very hard to keep everything in the mind. So only important things we are learning. You will see soon why we are too much concerned about bacterial cell wall. Because bacteria are the first prokaryotes that why they want to learn about their cell wall. The reason is they can cause disease to human. They can cause disease to other animals. Bacteria are considered as pathogen. Not all, but most of the bacteria we consider as a pathogen. Then we have it's important to human. They can cause disease. Then we have to learn how we can handle this situation. Then we learn cell wall. Right? Anyway, plant is after animal, the second important organism is the plant. So we have to learn the plant cell wall because they're producers. So the comparison is last, prokaryotes and eukaryotes, but we are getting little, the exposure to prokaryotes, and then which help us to remember things and which remember us to learn all these at the end, right? Okay, the major difference we said, prokaryotes, they don't have a nucleus. So you can see here, the DNA is called circular DNA. Why it is called circular DNA? DNA molecule is like a circle. The double helix, it's just like a circle. There's no ends. It's just a circle. But eukaryotes, they are straight lines. DNA molecule is like a line. And the bacteria, they don't have a big DNA molecule. That's a small DNA molecule. It's just like a circle. And it's not covered with the membrane. Cell membrane is not covered. No single or double, no membrane. And it has another name, nucleoid. We can use this word called nucleoid. The special name is given to such thing. No covering. It's a circular DNA. It's called nucleoid. Right here in this picture, you can see the cell wall. The outer layer is called capsule. And then the second one is cell wall. Third one is the yellow color one is the plasma membrane. Right? Cell wall, capsule, and plasma membrane. Okay, there is a separate unit again, microbiology. You can understand how far we have to learn about these microorganisms, but we take one by one, step by step to learn these things. Because we are learning all these things, it's repeating again, like bacteria structure again is coming up when you learn the microbiology. So it's okay to take some information from that unit and just add here. It's no harm because you have to learn that one. Right? Now, What is the composition of the cell wall of bacteria? What is important of this cell wall? Why it is important? Okay, the people, when they are concerned about the bacteria, they can cause disease. They are called pathogen, and scientists trying to find the uh, treatment. Scientists trying to find a method to prevent getting bacterial infection. Scientists trying to find a better medicine to bacteria. They did so much of effort in finding things. The very classic finding is a scientist called Graham. She used to color this bacteria using a dye called Graham staining is the his color. He used the Graham staining. He has seen one bacteria is a blue color, and sometimes he can also observe pink color bacteria. The stain give pink color and also a blue color. So he can take a sample from human, let's say from the throat, or maybe a urine sample, and then he can culture the bacteria in the lab, or he can take the sample and put the stain on it, and we can uh, check under the light microscope. So light microscope, still we can see after coloring this bacteria, like a tiny dots. And he has seen these two colors. So one is a blue color, other one is a pink color. And why this is important? Because if someone sick and is going to the doctor, now we have a better method. We have a better lab techniques. But years back, everywhere is not developed. But they can easily take sample and drop of this dye add to the slide, glass slide, and you can look under microscope. So they can see a positive. That means stained positive. That means it is stained. I call gram positive. Gram staining is positive. It gives the color. So it's a blue color. Gram staining doesn't give a color. So it's give the pink color because it's not a positive. It's a negative. Positive is blue color, negative is pink color. So then we can divide bacteria into two groups, gram positive bacteria, gram negative bacteria. So many years back, then the doctor can decide, okay, you are infected with gram positive bacteria. I should give this medicine to you. You are infected with the gram negative bacteria. Then these may be the bacteria. I should give this medicine to you. It's easy for them to decide the medicine. Now, why some bacteria get blue color, other does not get blue color? It depends on the cell wall. So then they have a different cell walls. Okay, here cell membrane. The bottom layer is a cell membrane. Cell membrane is common to all living organisms. So it is again made out of phospholipid. The phospholipid make two layers. The middle is the fatty acid, and the head is the phosphate group. Here they don't show the proteins, but protein should be also there. But for us, we are not going to learn cell membrane here, but first we identify cell membrane. Now cell wall is on the top of the cell membrane. And you can see cell membrane is made out of a special molecule called peptidoglycan. Peptidoglycan. And some textbook still they use old name called murine, but peptidoglycan is more common now. The word is very common, peptidoglycan. So we can say gram-positive bacteria cell wall is made out of peptidoglycan. Right? Cell wall is made out of peptidoglycan. This information is enough at the moment. So when we're learning further, we can add more information. So bacteria have cell wall. One group of bacteria called gram-positive bacteria, cell wall is made out of peptidoglycan. And the gram-negative bacteria, they have a cell membrane again, and they have a very small amount of peptidoglycan layer, like gram-positive bacteria. They have a small amount of peptidoglycan, and on top of that, there is a something called lipoprotein. Lipoprotein is lipid and protein. And then it has the fourth layer, outer membrane. That means it has inner membrane, cell membrane, outer membrane, and peptidoglycan and lipoproteins. So what's the difference positive and negative then? Positive has only a very thick peptidoglycan layer. Negative has very thin peptidoglycan and additionally lipoprotein and outer membrane. So it has three components: peptidoglycan, lipoproteins, outer membrane. Three layers. And here it's only one layer. It has only one layer, peptidoglycan only. Right now, some of the cell wall component, if you look at the cell membrane, the phospholipid is phospholipid, same in human, same in animal, same in plant, same in bacteria. But component of cell wall, they are different. That means we don't have in our body this kind of a molecules. So gram-negative bacteria, they have these lipopolysaccharides, abbreviated by LPS. So this lipopolysaccharide is toxic to human body. That means they can make some symptoms, it can make some signs in our body if the gram-negative bacteria is infected. That means cell wall component can make us sick. That's why it's important to learn the cell wall of bacteria. You should also remember, when you are learning something, it is very important because we are going to use that one. Now simply we don't learn cell wall of bacteria just to learn it has cell wall like this. It's important for us. It's coming up in the coming lessons, more about these things. Okay. Right, what you see here, it's a plant cell. You can see nicely the cell wall and the chloroplast. Green color one are the chloroplast. And you can see the thickness of cell wall. It's quite thick. Right. Plant cell wall is made out of cellulose. So we learn what is cellulose. Cellulose is a glucose molecule, and the glucose molecule is slightly different. It is a beta glucose molecule. So cellulose is made out of beta glucose. And we learn beta glucose molecule joined together to make the cellulose molecule. Beta glucose molecule joined together to make the cellulose molecule. Cellular molecule is a long chain. It's a long chain molecule. Then many cellulose molecule join together and they packed to make a layer like this. So you can see here this layer, this one. So assume this is the cellulose layer. And cellulose layer very tightly packed inside with other molecules. So other molecules are the matrix. That means it's a stage. First, the other molecules make the loose stage, and the cellulose molecule are tightly packing into that. So what are the other molecules? Other molecules are hemicellulose and pectin-like molecules. But the cellulose is the major component. Pectin and hemicellulose, pectin and hemicellulose are some other molecules we can find on this stage. And next to that, there is another layer. So what is the function of this yellow layer? Yellow layer attached other cell, attached with the other cell. So it make a glue, it make the middle layer between two cells. The two plant cells join together. In the middle, we can find this yellow layer. It helps to glue these two cells. So that is called middle lamellae. And okay, I think the wording is now cell. This one represents cell membrane. Yellow one is a cell wall. That's a cellulose layer, and the middle lamellae is this one. It's starting with the cell membrane, right? Cell membrane, cell wall. Again, it's the same story. Cell wall is made out of cellulose, and the middle lamellae is made out of calcium pectate. Calcium pectate. It's a glue structure which join two cells. And sometimes, some cells, the cell wall is getting very stronger. It's a very strong cell wall is created by lignin-like compound. So we have a separate section for plant. We are going to learn all these things: lignin, suberin, where is it, what is the function, which cells we can see them, which cell we can't. So you don't worry about certain thing too much at this case. Just learn it, okay, like that. Again, in detail, we are going to learn in plant. So here, what you have to remember, okay, plant has a cell wall made out of cellulose. If it is only cellulose, we call primary cell wall. The plant, some cells, they make a secondary cell wall with lignin. They're very strong. So what are the cells, where we can find in plant, why it is important, we're going to learn in detail. Okay, another thing is in plant cell, they have a connection between one cell to another cell. Not like animal cells, one cell connected to another cell through a channel. That's a small channel between one cell to another cell. That is called plasmodesmata. It's an opening. It's a complete opening of one cell to another cell. And then another place, the cell wall is very thin. cell wall is very thin, and cell membrane are two cell membrane. That means one cell membrane and other cell membrane are very close to each other. So they are called pits. Plasmodesmata is a connection opening between two cells. Pits are thinning of the cell walls. So they help for the communication. Again, we are learning what is this communication, what is the method of communication, how it's happening, like all the detailed method through plasmodesmata we are going to learn. Because we're going to learn there's a pathway called apoplast pathway. I don't even mention. Apoplast pathway is the movement of water within the cell. What is the importance of plasmodesmata and how it helps? All we're going to learn, right? So at the moment, you know, plasmodesmata is a connection between two cells in plants. Enough. Pits are thinning of the cell wall, very thin area of the cell wall, and cell membrane are very close to each other. They can better communicate. Pits and plasmodesmata. Okay, I think our time is. I can't see the time here. I'm going to stop soon because of this today the difficulty in connecting to the Wi-Fi and not in the